All right, this is the Chi-Square lab. I'm going to walk you through at least part of it. And the first page of the lab has a lot of good information to help you kind of see what I'm looking for, what is it that, what I'm going for. The main thing about this lab is you're going to do 3 activities. You're going to fill in this actual document and then save it. I would just add your name to the file title so you know that's the one you actually filled in. And then upload it to the same portal where you found the file on eCampus. All right, so we're going to do 3 activities. The first 2 is a little data collection and a little data analysis. It's just a simple data collection and data analysis. And the point of that is to demonstrate the concept of what we call the p-value. And then the third activity, you're going to look at a research paper that I found recent, it's pretty recent, that uses p-values, and you're going to be able to see how they made their conclusions from the p-values. It's not, the scientists are not making, conclusions are not just your feelings. You have to show the math. The math will tell you the data. When you do these statistical tests, it will tell you what the correct conclusion is. So it's not something subjective. It's not based on how I feel. It's the math will tell you the conclusion. So when I look at a paper, I'm looking at the math. I'm looking at their experimental design and such. But as a student, you should definitely be able to look at their statistical tests and verify that they made a reasonable conclusion based on that. And that's what we're practicing here. Did they make reasonable conclusions based on what the data analysis showed? Okay, so the test that we're going to use is called the Chi-Square test. It's not chi, although chi is something else, but this is chi, the Greek letter chi. There are other statistical tests that are used in different situations. You may know the student's t-test, one-tailed and two-tailed. You may know the ANOVA. But in any case, we're going to do a fairly simple one, and then all of them come up with a p-value. And the p-value, that always means the same thing. And it's always interpreted in the same way. So the choice of statistical test is not something we're going to address today. In your statistics class, you can address how you know which test to use. But when you get to that p-value, from that point on, the interpretation is the same no matter what test you're using. All right, so like I said, you're going to collect some data. You're going to flip a coin 50 times. And the probability of getting heads is a half, and the probability of getting tails is a half. Put in how many flips you got that were heads, how many flips you got were tails. Just go ahead and do it. Get some actual data. And then expected, the way you get expected is you multiply, in this case it's pretty intuitive, but you would multiply the probability of getting heads times the total number of coin flips, 50, to give you the expected number of heads. So if you expect to get 50% of the flips are heads and you do 50 flips, then you would expect to get 25 heads. And the same for the tails. And then across each row, you will determine what we call the deviation, O minus E, the difference. All right. So we're always comparing, we're always going to be comparing 2 groups. In this case, we're going to be comparing the observed set of data to sort of the perfect theoretical set of data that we call the expected. Those are going to be the 2 groups of data that we're going to compare. But there's always, in a statistical test, you're always comparing 2 groups, sometimes more than 2. But you're, in a simple statistical test, comparing 2 groups. So that's what we're going to model here. Then you're going to toss 2 coins 50 times. And you can, on a scratch sheet of paper, you can keep track. How many times do you throw them and you get 2 heads? How many times do you get any combination of 1 heads and 1 tails? And how many times do you throw them and you get 2 tails? All right. And then here it shows you how to work through what the probabilities would be for each of those outcomes. You can probably guess what it is. Your observed would be whatever you got when you actually threw the coins. And then that number should add up to 50. And then to get expected, you multiply the probability for that outcome, heads, heads, for example, times 50 to get the expected for heads, heads. The probability for any one heads and one tails combination times 50 to get the expected for that group. And the probability of tails, tails times 50 to get the expected. And in the expected, it can be a decimal. And let's leave 3 or 4 decimal places on any calculation that we do. All right. All right, so after you've gathered your data from your coin flips, we go on to do the actual chi-square test. All right. The chi-square test is called the goodness of fit test, where, like I said, you're comparing 2 sets of data. In our sample, we're going to be comparing sort of an ideal, perfect set of data for coin flips to an actual observed, collected set of data for coin flips. And we're going to compare them, and we're going to see if the groups have data there's always going to be a little difference between the data, but is that difference really large, really significant? Or is the difference between the 2 groups insignificant? All right. You start with the null hypothesis. Sort of the default hypothesis for any experiment is typically that there's no difference between the 2 groups. So let's say instead of doing coin flips, you're testing a new drug. This is pretty timely. You have a group that doesn't get the drug. They get a sugar pill, a placebo. You get another group that does get the drug. And maybe you're measuring, you have to have something to measure, so you measure, maybe you're testing a new blood pressure medication. And you want to see if the 2 groups, you're going to measure blood pressure maybe. So you give, the groups are taking the pill, and then they come in and have their blood pressure measured, and you collect all that data, and then you compare the 2 groups. Does the group that didn't get the drug have a different set of data for blood pressure than the group that did get the drug? You know, is there a significant difference between them? Is the drug having a significant impact on blood pressure? Or is the data pretty much the same in both groups, like the drug's not really doing anything? All right, so that would be one example. You can be comparing anything, but you're looking to see if the data, the 2 groups when compared, are significantly different or not significantly different. All right. So in this case, we're going to be comparing actual coin flips to a perfect predicted set of coin flips. So here's your H null, your null hypothesis. It's H with this little o here. The deviation or difference between the 2 groups' data is not significant. It doesn't matter what, that's always the null hypothesis in some form or fashion. You might say that the blood pressure measurements between the 2 groups is not significant. You can modify it for your particular experiment, but it's the concept is always that H null is that there's not a significant difference between the 2 groups. The other possibility, HA, the alternative hypothesis, is that the difference between the 2 groups' data is significant. Something is having an effect. You would assume in my example, it's probably the drug. If the group that got the drug has a significantly different result for blood pressure than the group that did not get the drug, and the only thing different about the 2 groups is the drug, then it's reasonable to say the drug is having a significant impact on blood pressure. So in any case, you're going to do the calculation. You're going to come up with what's called the chi-square statistic. So this is the chi-square statistic. It looks like it's the Greek letter chi and then a little 2. It's a little exponent 2. So chi-square. So you're going to be determining a chi-square number, and then from that you determine a p-value. And the p-value tells you whether or not you're going to conclude H null, or are you going to conclude HA? Are you going to conclude that the deviation between the 2 groups' data is not significant? Or are you going to conclude that the deviation between the 2 groups' data is significant? That's completely dependent on the p-value. It's not about your feelings. This is science. We don't care about your feelings. So the math will answer the question. So I'm going to walk you through this with the one set of data from the coin flips, and then you can do the other one. I have another chart down here for the 2 coin flips that you can do on your own, but I'm going to walk you through the first one with my set of data that I did. So I flipped 1 coin 50 times, and I got 23 heads and 27 tails. You'll get something else, but that's what I got. So you do yours later when you're doing this on your own. Do your own data. I don't want you to fill this in unless you actually did get this number. It could happen. Now what you expect would be 25 heads and 25 tails. I'm just looking at the top of each column and it's telling me what to do, really. All right, now the deviation between those two, 23 minus 25 is -2 on that first row. On the next row, 27 minus 25 is +2. So the sum of the deviations should be 0. That's kind of a self-check. And then O minus E squared, you just take whatever you had in the previous column and you square it. So 4 and 4 in this case, it's easy. It's not always going to be the same, but in this one it is. And then O minus E squared over E. So I take the O minus E squared from that row, 4, and I take the E from that row, 25, and I have my calculator because I don't like to use my cell phone calculator. I find it really annoying. And so 4 out of 25 is, I could have done that in my head. I don't know why I did that, 0.16. And then this one is 4 over 25, so in this case it's the same. So the chi-square statistic, which is down here at the bottom, right here in this little area, is, if you scroll up, this is the equation for the chi-square statistic. Chi-square equals the sum of, which is sigma, and then O minus E squared over E. So do you notice how we did O minus E squared over E? And then we're going to do a sum of all of those together. So you add down the column to get your final chi-square statistic. So in this one, it's 0.32. Okay? So that's your chi-square. So you have to find your chi-square. Then you're going to use a chart. There are some computer programs that you can put in your chi-square and it will tell you your p-value. We're just going to go we're going to kick it old school with a chart. So sometimes when you read like a journal article, they'll have an actual number for p. We're just going to use a chart, which is just going to get us in the range of a p. And what I mean by that, if chi-square is 0.32, then we're going to use this chart, and I provided it here for you. It's the same thing. This is a fixed chart for chi-square. So if you look this up online, you would find exactly the same thing everywhere, these same numbers and everything. All right, so on this chart, your p-values are across the top in yellow here. They're in yellow. Those are your p-values. You have to decide which one, though, matches with your data. Everything in white are your possible chi-square numbers. So remember, our chi-square number was 0.32. But the thing that's really going to make your life easier is that you don't use the whole chart. You only use one row. And the row that you use is called degrees of freedom, DF. DF is over here in blue. Now DF is how many groups do you have minus 1? So in other words, we have a group of heads data and we have a group of tails data. So we have 2 groups. And then DF is number of groups minus 1, so our DF is 1 in this case. So we're only allowed to use row 1. So I'm going to highlight it here, I guess. Okay. Let me see if I don't know if I can shade it. Let's see. Let me see. Okay, I'll give it an orange color. That's a little bit, maybe. Okay, so on this row, DF1, you got to figure out where, now in this middle section, this is where your chi-square numbers are. Now I shaded them orange, but now your chi-square number is 0.32. 0.3, all right, 0.32. So if I look at these numbers here, if you look right here, now I'll give this a different color, maybe a darker orange. Okay, you see right here where I find this chi-square number is 0.016, and this chi-square number is 2.7. And that's a pretty broad range in between them, but 0.32 falls in between 0.016 and 2.7. So you have to be pretty good with, especially with decimals, sometimes students get confused, but 0.32 falls between 0.016 and 2.706. So that means my p-value falls between whatever is in the yellow section up here. My p-value falls between 0.9 and 0.1. So that's what I'm going to put here. And you got to be careful with your greater than or less than. All right? Your p-value is between all right, so it looks like that. 0.1, p is between 0.1 and 0.9. And you may have to read it out to yourself. P is greater than 0.1 but less than 0.9. Kind of read each part of it. Make sure that makes sense. Don't put that p is less than 0.1 but greater than 0.9. That doesn't make sense. At least not in this case. All right, then state your conclusion. There is no option here. Your conclusion, and I actually wrote it down here, you got to scroll. All right, forming conclusions from p-values. This is it. This is not up to you. All right, any p greater than 0.05 results in the conclusion, quote, failed to reject H null. Now, we don't say accept H null. We don't say we proved H null, but that's kind of the direction we're leaning anyway. We never say proved it, but we say we didn't rule out H null. All right. So in that case, our p is greater than 0.05 because it's greater than 0.1. And so our conclusion is fail to reject H null. All right. Now, like I said, H null said that there was no significant difference between the two groups of data. So what this means is that for our data, what we observed is not significantly different than what we expected. So chance, the coin flipping matched within a reasonable expectation the expected data within a little bit of error. That the amount of error is just small and acceptable. All right. Now, what if we had gotten if we had gotten a chi-square value that was quite large, and we went down to the chart, let's say, and let's say we had gotten something, maybe our chi-square number was more over here, then our p-value would have been something like this, 0.005. In that case, we would have said, instead of fail to reject H null, we would say reject H null and favor HA. That's what we usually say. So you reject the default hypothesis in that case, if that had been the case. And you say reject H null in favor of HA. It means that you believe that the deviation between the two groups' data is significant. Well, not that you believe, but that the statistical test shows that. All right, so you're either going to, you can use the same language. You either fail to reject H null, that's if p is greater than 0.05, or if p is less than 0.05, reject H null in favor of HA. All right, so on the data that I got with the chi-square number of 0.32, this would be my p-value from the chart, and then this would be the corresponding conclusion. So you're going to fill in your observed and expected data for the two coin flip. In this case, you have three groups of data. You have the two heads group, the one heads and one tails group, and then the two tails group. So your three groups, your degrees of freedom will be one less than 3, so 2. So you will use the line on the chi-square chart marked DF2. You'll determine your chi-square statistic. You'll go to the chart and see what p-value matches with that, and it'll be a range. And then based on what I've written here, you come up with what the corresponding conclusion is. So the data, the calculations, and the conclusions are all set. It's not, it's not up to the individual scientist to decide what is significant and what is not significant. So that's what, we agree on these statistical tests. We're all using the same concepts here. All right, so you will do that. All right, now the last activity, I have posted this article here. And I give you a certain amount of background, but it's involving genetics and it's involving pharmaceuticals. So it's a drug. It's actually a drug that was developed, you may recall from chapter 7 that I was telling you about CCR5, that co-receptor that is needed for HIV to enter cells. Well, they were able to come up with a drug. Pharmaceutical company came up with a drug that, I think it was Pfizer, came up with a drug that blocks that receptor in healthy patients, and it helps patients, slows down the progress of HIV. But one thing they were worried about was that your liver breaks down drugs. And there was some reason to think that certain genotypes break down the drug faster than others. And that would be important to know because you would need to give that kind of individual more of the drug, probably higher dose. There's all kinds of implications medically for that. So I'm having you kind of look into that just a little bit, okay? Just doing a little poking around. But the article, you'll just answer these questions. I give you some links to go to, and I give you enough background for you to be able to answer these questions. The document is, the paper is, I think it's Vorvehis. I'm not sure how to pronounce it, but I'm calling it Vorvehis 2019. And here's the paper. And you can read the title, and after you've read the background information that I gave you, I think you'll be able to figure that out. And then I really direct you mainly to a couple of the data tables. So table 1, I think I asked you to look at figure 1 and table 2. So here's figure 1. Kind of takes up more or less of the whole page, half the page on page 146. And then table 2 here on page 147. And so here you can see the p-values for the different groups that they were comparing. So you'll do a little figuring out of what some of these things mean, but ultimately you're just seeing p-values between comparisons of two groups. And what they're measuring, in the example I gave you before, would be like blood pressure between a control group and a group that got a certain drug. What they're measuring actually is how fast is the Maraviroc being broken down in the blood, so plasma levels of drug in a control group and in groups that have certain genotypes. So I think you'll be able to figure this out if you're having any trouble, but you don't have to read the whole paper. You're going to be directed immediately to the title of the paper, figure 1, which is here on page 146 of the paper, and table 2. And figure 1 and table 2 virtually show the same data, just in two different visual ways. And they have some definitions down here in the figure legend, so make sure you read that a little bit, skim through that and see if you can figure that out. But I think you guys will be able to figure this out and see how these p-values are used and why the p-value matches, in this case, the title of the article. The title of the article tells their results, tells their really their conclusions. But so I think you're going to enjoy that. I think you're going to see a direct value, no pun intended, of understanding what p-values are from a practical point of view, so that you can interpret data yourself and then, make sure that, scientists look at these things and make sure that do I agree with their conclusions and that kind of thing. So you'll spend a little bit of time, but everything you need is, like I said, in just a few places in this paper or in the worksheet that I've provided for you. Okay, so I hope you enjoy that. I don't think it's overwhelmingly difficult, but like I said, give me I've just developed this activity, so if you have any problems, let me know.